webinar today. I want to definitely thank Arjun and Lisa Kozlowski and the JPA for arranging this. So as Arjun has already mentioned, I am a Jeff alumni, and I recently finished a postdoc in cancer biology before I started my current role at Miltenny. And I have been with Miltenny for almost a year, but I've literally been where you all are. And so I'm really excited that I still get to work with you in my current role. Today, we're going to discuss the current state of antibody production and why having consistent synthetic protein binding reagents are crucial to generating reproducible data, particularly in a world where a lot of the validity of scientific data is called into question. And we are dealing with quite a reproducibility crisis. I'm going to focus a little bit on the use of antibodies for cell analysis, particularly flow cytometry, but a lot of these principles I think can apply to imaging, microscopy, and in general, I think it's going to be really relevant to any of you that routinely use antibodies. And as a note, I cannot see where the chat feature is, so if you have a question, I think I have plenty of time at the end, or if you're shy, I also am dropping my email in here, so you can ask me that way as well. I'm going to start my talk today by giving you an overview about the reproducibility crisis, what that means, and how it's evolved over time. And I think I'm going to dedicate this talk today to anybody who's ever tried to reproduce a published experiment without success, or even if you've tried to redo an experiment that somebody in your lab previously did, because we all know how frustrating that is. Then I'm going to do a bit of a history lesson and a lesson in basic antibody biology. I'm going to explain what recombinant antibodies are and how they can help us improve reproducibility, essentially how these aren't your mom's monoclonals. Then we're going to discuss the validation steps that we utilize in-house at Miltenny. And at the end, I'm going to give a little added information on how we're participating in the greater scientific conversation around antibodies. And then, of course, what you can do as a scientist. As a disclosure, as already mentioned, I do work for Miltenny. So in particular, I'm going to be talking about how we specifically produce these recombinant antibodies, but I'm going to try to give a really general overview of antibody production and the issues that people face when working with these materials. So I show this slide kind of to let you know this is where I work, not here precisely. This is our German headquarters. I live in South Philly, but I'm going to show this slide also because it kind of relates to our topic today. Miltenny Biotech was founded in 1989, and from the very beginning, it was our goal to provide high-quality products. We've always had this huge push and focus on R&D. A large chunk of our employees still work in R&D, and we churn out a lot of scientific publications. And we started antibody production in the early 90s for our microbeads. So while we're known for being a magnetic cell sorting company, the 30 years of success from Miltenny really sprung out from our antibody manufacturing from the very beginning. Here's actually a picture of Stefan Miltenny, who's a strange man and a colorful genius. And this schematic drawing of our columns is a figure from Stefan's 1989 paper where he describes the origin of MAX technology. So you'll probably recognize these columns here that we are using for magnetic cell separation. Stefan used to make these in his mom's basement using her washing machine as a centrifuge. And for this talk, I'm going to spare you those very interesting details, but I've included this figure to show you from the very beginning, we've been intimately linked to antibody production because Stefan knew really good antibodies were crucial to ensuring the magnetic labeling of cells as well as the cell analysis, which he was using flow cytometry. So I'm going to start today by, I think, probably scaring the living bejesus out of you. We're going to talk about reproducibility in science and how the reproducibility crisis has been unfolding now for over a decade. So over the past several years, there has been a growing concern within the scientific community that a lot of published results are not reproducible, and antibodies are considered one of the biggest variables in the system. Here is a 2008 study by the Human Protein Atlas Project. They found that less than half of the commercially available antibodies actually recognize their targets, which is troubling. This is a picture of David Rim. He was a pathologist at Yale in 2009, and his lab had this, I think, really devastating experience. So they were on their way to characterizing a new cancer marker But they realized that a new batch of antibodies they were using to detect this marker was failing to give a reproducible staining, and it ended this $1 million biomarker development project. And then there's this study in 2012 by Amgen, and they revealed that the seminal findings of only 6 out of 53 hallmark cancer papers were actually able to be reproduced by Amgen scientists. And this led to this debate you've probably heard at this point as to how rigorous the guidelines for authors should be and whether maybe more transparent reporting is required. So in 2014, the NIH met with leading publishers in order to try to find some sort of solution for this problem. And we see in the subsequent years, there's been all of these publications coming out of Nature calling on the scientific community to create and then use recombinant antibodies that have a defined DNA sequence. And then most recently, in 2018, a group explored 185 hybridomas chosen at random. They found that roughly 2/3 of them were producing antibodies with alternate heavy and light chains, which obviously is going to affect the specificity as a protein binding reagent. And then again, in their conclusion, they called for the use of recombinant antibodies in both research and diagnostics. Here's a figure that I love. This is from a Sigma survey in 2014. They published this survey on translational research. They asked people what products are causing the most issues with your reproducibility. So right after animal models, which are obviously going to be very unpredictable, the scientists voted for antibodies. So the fact that antibodies took this number 2 slot really shows that we're aware of this problem. Scientists know antibodies are a very significant variable in this system that lead to unreproducible results. Here's another really disturbing figure. This was from Nature a few years ago. They estimated the financial loss of purchasing these, like, quote, bad antibodies. They estimate half of all antibodies purchased are unvalidated, and therefore in the United States, for a total spend of $700 million, $350 million is just wasted. This also is applicable globally. So they estimate $800 million wasted on bad antibodies. So what are recombinant antibodies? What are these antibodies people are asking for? How are they going to help mitigate reproducibility issues? And so I thought it would be helpful if I go and do a quick kind of overview of antibody structure. An antibody is comprised of 2 heavy chains. Those are shown in this figure in green, and 2 light chains here in blue. So both the heavy chain and the light chain contribute to the variable region of the antibody, or the FAB, and therefore they both contribute to the antibody's binding properties, or specificity. And then the tail end of the antibody, shown here is this FC portion. This stands for fragment crystallizable. This is the constant region. This is the business end of the antibody, I would say. It directs the immune system by binding to FC receptors on immune cells in a lot of cases. So, for example, the variable region would be bound to a pathogen, and then perhaps a macrophage will come along and bind to this constant region of the antibody, and it will be activated to be able to engulf that pathogen. The different types of constant regions have different roles in a variety of immune functions, and these are called isotypes, and there's even sub-isotypes of these different structured antibodies. So the last point to consider is that each individual B cell will secrete a unique antibody containing a unique variable region that recognizes one small portion or epitope of a surface marker. This is what we mean by clonality. And kind of ironically, I think, while the variable region for the most part will stay the same, the constant region can actually change through the lifespan of a B cell during its natural maturation and activation. So that brings us to what I'm calling a short history of antibody manufacturing. For a very long time, pretty much as long as immunologists recognized what antibodies were, we have been trying to make them to be used for either protection against various pathogens or for use as a lab reagent. And antibodies were originally and still are manufactured by immunizing an animal, such as a rabbit, lots and lots of rabbits, with your antigen of interest. then the antigen-specific antibodies are purified from the blood. So therefore, we end up with this collection of many different antibodies from many different B cells, all with different variable regions that bind different epitopes of the antigen of interest. So thus the term polyclonal, or I'm calling it antibody version 1.0. So as you can imagine, we're recognizing lots of different epitopes on the same antigen, so we're going to get quite a strong signal, but it's a really inconsistent research tool. So imagine this, you have one batch of antibodies that are finished, so you got to go out and get yourself some more rabbits. So let's hop forward to the 70s now. Disco is really big, and Kohler and Milstein published this figure in a paper in 1975 where they described the creation of monoclonal antibodies by hybridomas. So let's call this antibody 2.0. These hybridomas are clonal, so you can generate theoretically a single kind of an antibody with a known specificity continuously. So to create these hybridomas, the animal is immunized and its splenocytes are collected, and then they're fused with a myeloma cell, and a myeloma cell is just a malignant B cell. And to do this, they use a selective media called HAT media. And in this media, anything that isn't a fusion of these two cells, this little baby Frankenstein cell, it will die. Then essentially you can just dilute these until you have one cell in each well. You can expand them, you can screen your antibodies, determine which is your best clone, and then you just culture that as a new cell line. And these hybridomas are immortal because of the nature of that myeloma cell. And they're just these continuous little antibody factories, each producing its own unique antibody. And so obviously this was a huge step forward, like a really drastic improvement on specificity and consistency. And it's just way better for relying on a nice, consistent product. But these hybridoma-derived monoclonals also have major drawbacks, and this kind of sits squarely in the center of our debate surrounding reproducibility. So let's kind of compare these tools, version 1.0 and 2.0, and then we can start discussing our steps forward into producing recombinant antibodies. So just as a little bit of a review here, polyclonal antibodies, again, remember, they're a mix of these antibody clones and isotypes. They have differing specificity and sensitivity because they're made in an animal. It's just part of its natural immune response. So they can provide you a very nice signal because they recognize a lot of different epitopes, but their consistency from lot to lot is quite poor. So in a lot of instances, they're not a great tool for cell analysis. So monoclonals, version 2.0, this is a mixture of homogeneous antibodies. They recognize a single epitope for a very high degree of specificity, and they can be more consistent. But again, we're looking at a mix of heavy and light chains, and this can contribute to problems with reproducibility. So you can see that the natural progression is here to improve upon antibodies as a tool for research, to create something with a defined content. And that's what we've done at Miltenyi, specifically with our flow cytometry antibodies. And I'm going to talk about how these are created in more detail on the next slide. But essentially, we're taking the existing recombinant technology and applying it for the first time to manufacturing flow antibodies on a commercial scale. So essentially, we take our favorite B cell clone, we sequence the variable region, we clone it into a construct and produce them in a stable mammalian cell line. And what you end up with is a synthetic sequence-defined protein binding reagent, which is way more consistent lot to lot. And these have really exceptional sensitivity and specificity, and it is a vast improvement on hybridoma, which is why I say they aren't your mom's monoclonals. It's a revolution. So let's look at our process side by side. As we saw earlier, when we're creating a hybridoma, the variable region, the crucial antigen binding region is made up of both the heavy and light chains. So hybridomas are this fusion of the B cell clone you want combined with a malignant B cell. So this myeloma cell is going to continue to produce its own heavy and light chain. Therefore, a hybridoma-produced monoclonal antibody is going to have these multiple heavy and light chains commingling, and that can create unwanted antibodies. Also, remember what I said, they did this in the 70s. A lot of these clones have been around for decades. Over a long period of time, and a lot of these passages, spontaneous mutations can occur. This can lead to genetic drift. And the final product can also be contaminated from immunoglobulin from the sera required to culture a hybridoma. So you have this completely unrelated antibody as well as your mixture of heavy and light chains. And recombinant antibody manufacturing resolves all of these problems. And that's why I think we're seeing a lot of the therapeutic antibodies popping up in clinical trials now. They're manufactured in this way. They're not produced in B cells. So you don't have to worry about contaminating heavy and light chains. Essentially, as you can see in this figure, we've cloned a defined sequence into a mammalian cell line, and then the mammalian cell expression system is programmed only to express the product of our DNA sequence. These are also cultivated in a defined culture condition, so we're not using serum here. So this reduces the possibility of further antibody contamination. So all of this creates super high lot-to-lot consistency in a defined synthetic protein binding product with very high purity. So for example, a 30 microgram vial of a recombinant antibody is naturally going to be more potent than a 30 microgram vial of a hybridoma produced monoclonal. And what I think is really exciting, recombinant engineering introduces the option for further engineering and optimization. So for example, I'm going to talk a little bit about how we've optimized some of our recombinant antibodies for flow cytometry in a moment. I think Miltenyi has really helped drive the industry in this direction. All the way back in 2010, we recognized the need for these recombinants and we started building our manufacturing platform. We launched our first recombinant antibody in 2012. We were the first provider for recombinant antibodies in flow cytometry. In 2015, we tried to expand the markers that we made. And 10 years out, we have the largest portfolio of recombinant antibodies optimized for flow. We call our recombinant antibodies reaffinity, or I may say today, I might call them RIAs to abbreviate. I have to say crazy words like Miltenyi, so my mouth sometimes needs to just say RIA. So I thought we could take a look at how Miltenyi has specifically tackled some problems with flow cytometry through some features of our RIAs. Here is a nice cartoon model of an antibody. And we already did discuss the structure of an antibody. So in orange, you're going to recall that this is the antigen binding site, which is derived from our hybridoma clones that have already been validated for flow cytometry. So this means our reaffinity clones have a similar epitope pattern as already known clones coming from hybridomas. And as I mentioned before, Miltenyi has been in the antibody manufacturing business for 3 decades, so you can expect that all of our antibodies are thoroughly tested for specificity and sensitivity. I'm going to talk about how we validate these a little bit more, but first I want to mention some of the other modifications that we made to our antibodies. Another change that we decided to introduce to these antibodies is that they are all human IgG1. So you remember I mentioned there's a lot of different antibody isotypes. Well, we have simplified this system. We've engineered them all in the same backbone, this human IgG1 backbone, which makes using isotype controls way easier in your experiments. So if you're doing your flow cytometry controls the correct way, you know that you should be using isotype controls. So you need to have an antibody with the same isotype that's conjugated to the same fluor as the antibody that you want to compare it to. So on the left here, we have 5 different antibodies for say, this is our panel for our flow experiment. And these are 5 different isotypes. So we're going to need 5 different isotype controls, and we need to have the right color for each of them. So this usually results in having a really large library of rarely used isotype control antibodies that kind of pile up in your refrigerator. Like, how many of you have that really intimidating and very annoying box of mystery antibodies that's been in your fridge for who knows how long? This is what that is. If you're using the RIAs, on the other hand, our recombinant antibodies, all of the isotypes are the same. So it doesn't matter if you have an anti-human, anti-mouse, anti-rat. It doesn't matter what your target marker is or what species it is. You're only going to need to have one RIA control and in the color of interest. And so this will make your experiment planning a lot easier, but I really just like that you can throw out the mystery box. Another thing about that human IgG1 is it's widely used now in generating therapeutic antibodies. So it's been really well characterized, and it's something that it's easy enough to mutate. So as I mentioned before, the constant region of an antibody is the business end. That's what immune cells bind to through their Fc receptors. But when you use antibodies as a tool in research, that binding can contribute to unspecific signal. So all of our RIA clones have this specifically mutated human IgG1 Fc region that abolishes their binding to Fc receptors. So this is going to reduce background noise pretty efficiently. I mentioned before, normal antibodies will bind to cells through this Fc part of the antibody, just as they do in nature. But how specific will the staining be now in your flow experiment if you have maybe some macrophages hanging around, or maybe you have some tumor cells? Anything that's going to have these Fc receptors is going to cause a false positive and really unreliable results. So what people do is they use a reagent called Fc block. And this is going to occupy that Fc receptor and will prevent this errant binding from happening. The issue with that, though, is Fc block is just comprised of other antibodies specific to the Fc receptor, and this blocking could be incomplete. So as a consequence, you could potentially be getting a false positive signal. Also makes it really challenging to appropriately set your flow cytometry gating and then interpret your data correctly. So the point mutation that we engineered into the reaffinity antibodies completely prevents that binding. So you'll have no background and you'll have much more reliable data. I want to show you what that looks like. So we tested our RIAs on different families of Fc gamma receptors, and we found that our recombinants virtually abolished the source background staining. So we're showing it here with some ELISA data. We're comparing a monoclonal, that's in that dark purple, and the reaffinity antibody in a light purple. So you can see for all of these Fc receptors, there's a virtually non-existent background signal from the recombinant antibody. And let's look at some customer data. I think that illustrates this point pretty nicely using flow cytometry. So here we're looking at CD4 staining on that y-axis, and we're looking at CD8 on the x-axis. So in columns A and B here, you can see two different antibody clones that are different for these different markers. And so these two plots show staining without the use of Fc block. And you can see, first of all, how drastically different the staining looks with these different clones. But you also see something really weird, which is what looks like this CD4 dim population. That is not a thing. That's not real. That is background staining. So now they've applied an Fc block, and you can see that we've reduced that background quite a bit, but it's not really complete. So let's take a look now at staining with our reaffinity antibody, our recombinant antibody. All of the background here is eliminated, and you can see they've done this again with Fc block, and it's no different. So this is kind of nice. It really reduces your need to have to do that step at all, saves you a little bit of time. And it also saves you that kind of uncertainty whether or not my signal is real, having to think about that. Now you don't have to. So now, as I promised, I want to give you an overview of how we validate our antibodies at Miltenyi. Throughout the past year and moving forward, we are building upon and extending our in-house validation process. And as new techniques become available, it becomes easier to thoroughly test antibody specificity, sensitivity, and reproducibility. And I think what's great is we want to show you in a transparent way how we develop, validate, and control our antibodies. And of course, this is a lot of additional tests and data that needs to be performed and processed. So, we've been slowly rolling this out, and you can see this on our product pages as more data becomes available. But the goal is to have this available for every single one of our antibodies. I'm going to call these the 3 pillars. So when we're trying to validate antibodies in a manufacturing setting, we're going to look at 3 things. The first is reproducibility in terms of lot-to-lot performance of the antibody. The second is specificity, which addresses the antibody's ability to recognize the appropriate target. And the third is sensitivity in regard to the antibody being able to send a significant signal to be able to detect the target. So let's talk about ensuring reproducibility first. Earlier, we kind of touched on the idea of contaminating heavy and light chains, and here we can see that in action. So in column A, you can see mass spec data of a traditional hybridoma, and the pink star represents a peak that is the light chain that comes from the myeloma fusion partner, and the green peak is a signal from the actual chain we want, the light chain from our B-cell clone. So not only can you see that we have contaminating chain from myeloma, but you can see in the difference between the top and the bottom in that column, you can see it's different for different lots. And that's really going to have a lot of lot-to-lot variation for us that could potentially even ruin our subsequent experiment if we're trying a new lot. So you can see in column B, where we have our recombinant antibody, our RIA, there's only one chain. It's only the chain that we've engineered our cell line to express. It's only the productive light chain. And in one lot to the next lot, it's going to be the same. So we have quite nice consistency there. So I've already shown you we're using this high quality starting material, only the information that we've encoded the cell to make. Our recombinant clones are more pure for that reason, but we also are using quality controlled fluorophores. And our antibodies are tested for lot-to-lot consistency at 2 different stages: after raw material production, before we do any conjugation, and then again after fluorochrome conjugation. So after conjugation, the antibody fluorochrome mixture is purified, which will remove any unbound fluorochromes and any antibody that's not bound to that. And since we use a chromatographic method to purify antibodies, any contaminating immunoglobulins would not be removed at this step. So it's really important our antibody product doesn't contain antibodies that are created by or with the fusion partner. And it's not possible to do that with a recombinant antibody. So the final product is then functionally tested on primary samples. And what's really important here is you have to use the correct conditions. So for example, if this is a marker that only shows up on a cell that's been stimulated in some way, that is the kind of cell that we are using. So it's really important to control for these. And every new lot performance is compared to previous lots by flow cytometry, and then the titer is adjusted accordingly. So what's really, really nice, I think, is we recommend an actual starting dilution for you for your experiment. So if you go by that, there's not going to be any need to titer when you get a new lot. So here we're looking at what that looks like. We're looking at human PBMCs from a single donor, stained with CD56 antibodies, conjugated to 4 different fluors. That's the only difference in these charts, is the fluorochore. So for the 2 different production lots we're looking at here, we're looking at lot A and lot B, and you can see, maybe you actually don't see, but it's perfectly overlaid. These histograms show you that their performance is virtually identical. So these are the main ways we're ensuring reproducible antibody manufacturing. Another really important aspect when dealing with antibodies is, of course, specificity to your target. If your antibody isn't specific, or maybe it was specific and the specificity isn't maintained, your entire experiment can be messed up. I'm thinking back to the RIM Labs project where they spent $1 million and it turned out their antibody was not specific. So all of our antibodies are tested in-house. And we do these specificity tests in a lot of various ways, but technology has become more advanced and easily accessible for us. So we're constantly trying to improve this QC and validation. On top of all those normal validation processes, we're also now testing antibody specificity using CRISPR-Cas knockout cell lines. So once our target marker is established, the cell line expressing the marker of interest is going to be found by high throughput screening. Then the target gene is knocked out in a suitable cell line by site-specific nucleases, and then single-cell cloning is performed to achieve a pure knockout cell population. So from those cells, genomic DNA is isolated, and the knockout is confirmed by sequencing of the target locus. And then the clones used for testing will be selected by the DNA sequence that has the most reliable knockout. And then the knockout cell line is compared to the wild type cell line by staining with the antibody that we've developed by flow cytometry as well as fluorescent microscopy. And this is what the results generally look like. So the antibodies are considered to bind specifically to the intended epitope if no antibody binding is detected on the knockout cell line. So for flow, we've generated this histogram, and we can see a positive peak for our wild type and a negative peak or no staining in our knockout line. And the same cells are analyzed by microscopy below. So you're seeing the signal only in the wild type cell line. And our antibodies, we kind of gave them our own clone names indicated by RIA followed by a number. And obviously, users aren't generally familiar with our clone names if they're switching away from a monoclonal. But we do want to give you as much information about how they compare to the traditional clones. So to do this, we perform competition assays to tell you whether or not they stain the same epitopes. So regular staining here is shown on the top as kind of a benchmark for this kind of experiment. And then what we'll do to compare is we'll incubate cells with pure unconjugated reaffinity antibody, and then we'll label after that with a hybridoma clone conjugated to a fluorochrome. So there's two potential outcomes in this kind of assay. So first, if there's 100% staining from this, then the hybridoma is recognizing a different epitope than our recombinant antibody. However, if there's no staining, this means that they have recognized the same epitope and our reaffinity antibody blocked the epitope. And this is how we display what that data looks like on our website. So we're trying to add these to each of the clones that we have created. So the plus sign here means that it recognizes the same epitope, there's overlap, and the minus sign means they recognize different epitopes. So our final pillar of validation, that I'm calling these pillars, is sensitivity. So regarding sensitivity, we compare our antibodies to commercially available and established hybridoma clones. So here's an example of one of these comparisons. And here we're looking at PBMCs stained with our CD56 recombinant clone, our RIA, and two other hybridoma-derived clones in the two columns to the right. So the upper row shows FITC conjugates, and the bottom are all conjugated to PE. So you can see our recombinant antibody on the left shows a really nice resolution. Look at that, the separation between the positive and the negative peaks is really nice. So you could see it has a higher sensitivity to the target compared to the other commercially available clones. So fantastic that we're doing all of this, but where can you find the info? As I mentioned before, we can't provide it all at the same time because we're not the largest company and we're generating these as we can. So we've been rolling these out as we can, and it's soon to be on every product page. And so if you see on the left-hand side, you can see our competition assays. That is on most antibody product pages. And we're rolling out the specificity and sensitivity data as we go along. I'm seeing them on more and more of the clones that I've looked into. One of the other things I really like is we're trying to post whether the antibodies can be used on fixed cells or not, which is something nice to know if you like to fix your cells before you do your staining. So I just want to summarize how our reaffinity or our recombinant antibodies can help ensure reproducible flow results. First, these are designed for flow cytometry. The antigen binding site is derived from a hybridoma cell line that was already validated for flow cytometry. Then we have eliminated the ability for these to bind to FC region. They're also produced in a standardized way. These are sequenced, and the conditions that the cells are cultured in are always standardized. Then we purify. Actually, we double purify. We make sure that by using chromatography, we have nothing that is either an unbound fluorochrome or an unbound antibody. We make sure it's just what you think it is. We have very well-defined lot-to-lot QC criteria, and we test each batch against the previous batch. And finally, we're trying to make this validation available to the scientific community. We're trying to be transparent, and we're trying to do as much extended validation of antibodies as we possibly can. And in the last few minutes here, I just want to give you some additional information on the topic in general and some of the ways that we're trying to contribute to the scientific conversation about reproducibility. So we, as the royal we, Miltenyi, have been involved in several activities connected to this topic. For example, we sponsored the second and third International Antibody Validation Meeting hosted by CITAB. Remember when you could have meetings? That was good. Not only did we sponsor the Antibody Validation Initiative of GBSI, we also actively participated in the workshop on how to ensure reproducibility in science. There's this fantastic BioCompare documentary. We took part in this. If you reach out to me, or maybe I can send a link through the JPA, and you should watch this. It's a free video. It's not too long, and if you run out of Netflix, it's definitely worth the watch. Lastly, we're constantly publishing papers, presenting posters at global conferences, and trying to give as much insight as we can into our in-house R&D. We are also really proud of our technical support, and I include myself in that category. We can help with questions related to our antibodies and products, but I'm also really happy to help with planning experiments, or if you have any issues with panel design and that's something that you want some help with, that's what I'm here for. And also please tell us if an antibody is not working properly. We can either get it to work properly, or we would certainly want to know if there's something wrong with it to prevent other people from using it. So that's what we're doing at Miltenyi to help, but what can you do as a scientist? Well, first, you can make your job a lot easier and more straightforward by purchasing recombinant antibodies. It's time to stop using your mom's monoclonal. Even if Miltenyi doesn't make one specific for your favorite marker, that's okay, but please use one from someone else, but it's time to switch. Use antibodies that have been validated for your application. You can do your own validations to ensure an antibody works for your specific experiment. Here's a hot tip. If you don't know if it will, or the website doesn't say, you can ask your rep for a sample. Most of us will say, fine. Share your methods as detailed as you can, as transparent as you can, so other scientists can try to reproduce your experiment successfully. Nobody wants to hear that, oh, so-and-so tried your experiment and they couldn't get it to work. Nobody wants that. So here's a really good example. I love this. This is the STAR methods, which was introduced by Cell Press for their articles. I think it stands for Structured, Transparent, Accessible Reporting. So if you haven't heard of this, it's really worth looking into. Use their guidelines for your publications. You can also, here's RRID, a reagent ID repository. These are just online databases with products that you can really easily cite in your experiments. You can rate antibodies that you use on platforms like BioCompare. It's like Yelp, but for scientists. And you can use search engines like Citab or Antibodypedia. And like I said before, report bad antibodies. If something is garbage, please tell the vendor so. Nobody wants to try to use something if it could have been prevented. And on that note, I want to invite you to join the flow revolution. I think it's time to rethink using monoclonals and upgrade to a more defined, consistent recombinant antibody. And so I put my email here. You can reach out to me directly. Just remind me that you attended the seminar, and I'll let you try a recombinant antibody of your choice. And if you want, I'd love to share that BioCompare documentary link. I think it's definitely worth a watch. So I hope this has been informative to anyone. I think we've all been there where we've tried unsuccessfully to reproduce something, even that came out of the same lab. And it's something that affects us all, this reproducibility crisis. So I think it's going to be scientists dedicated to raising our standards that can help revolutionize what we know and understand. So with that, I just want to thank you all for your attention. Thanks again for having me. Thanks to Arjun for putting this together. And I'm happy to take questions, or if anybody wants to tell me about a struggle that they had reproducing data, horror stories, anything like that.